I have a joke for you. Are you ready? Okay. What is the best thing about being 110 years old? No peer pressure. Ha 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 ha. All right. Welcome. This is going to be the meiosis portion of the cell division unit of mitosis and meiosis. Of course, you know that what you're looking at here is sperm cells, all right, trying to fuse and break through the envelope of a female egg cell, all right? Now, the crazy thing is you could imagine what would happen if a sperm cell with a full set of DNA, all right, fused with an egg cell also with a full set of DNA. The resulting cell would have twice the amount of DNA that a normal cell will have. All right. Now, what we know is that human somatic cells, every cell in a human other than the sex cells, they have 23 pairs of chromosomes with 46 chromosomes altogether. All right. So 46 total chromosomes. If this cell was made in a somatic fashion, this cell would be what we call diploid. It would have 46 chromosomes. All right. If the egg cell was made as a somatic cell is made, it would also have 46 chromosomes, and when they fuse together, we would have a cell with 92. All right. So, in order for the process, this process here is called fertilization, when a sperm cell and an egg cell fuse to form a zygote, is called fertilization. All right. Now, in order for fertilization to occur correctly, the sperm cell and the egg cell both need half of the amount of DNA that they would normally have. So that when one of these sperm cell actually gets the golden ticket, all right, and gets to fuse with the egg cell, that the DNA that they're fusing together between the two cells is equal to what a normal somatic cell would have. Now, that zygote now would have the normal pair of chromosomes being 46 total. So in order for this to work, the sperm cell needs 23 total chromosomes, one of each kind, one of each of 23 chromosomes. And the egg cell also needs 23 chromosomes of the 23 types. Now they both have 23 chromosomes each, and each of them have the same kind, not the same exact one, but the same kind of a number 1. They both have a number 2, they both have a number 3, they both have a number 4, and a 5, and a 6, all the way up to 23. All right. Now, these chromosome pairs are called homologous chromosomes. So what you're looking at here is a karyotype. A karyotype is a picture or representation of all the chromosomes in an organism. All right. So what you're noticing here is that for chromosome number 1, there are 2. Each cell in your body, except a sex cell, has 2 number 1s, 2 number 2s, 2 number 3s, 2 number 4s, 2 number 5s, and all the way down to 22. So chromosome number 1 through 22 are called homologous chromosomes. All right, they have the same exact information on them. All right, except the types of information they have are different. So if chromosome number 1 had 1 gene on it, which was for hair color, this chromosome number 1 would also have the same type of gene for hair color, except one of these might have information for blonde hair, and one of these might have information for black hair. They may both have the information for the same color hair, but that's not always the case. Sometimes they do, sometimes they don't. So what we mean by homologous chromosomes is chromosomes that have the same genetic types of information on them, but they don't have exactly the same information per se. So all the chromosomes up till 22 have the same genes on them, but those genes may have different forms called alleles. All right. The 23rd chromosome actually there's an X and a Y. These are not homologous, meaning they are not the same. All right. The two possibles are double X or XY. If you have 2 X chromosomes, you are a female. If you have an X and a Y, you are a male chromosome. You are a male. All right. So the information for maleness is on the Y chromosome. So the crazy thing is generally, if you have two X's, you are female. All right. In order to override some of the female types or some of the things that make an organism a female, you need a Y chromosome in order to override some of that information. All right. So you can imagine, if we go back here, the sperm cell has one of every one of these, and an egg cell has one of every one of these. So when they fuse together, to form a zygote, they end up having the full number. All right. So that's homologous chromosomes. All right. So homologous chromosomes are chromosomes which have the same type of information on there, but they may not have the same exact information. Now they could have the same exact information, but they're not likely to have the same exact information. One of them comes from one parent and the other one comes from the other parent. So that of your 23 chromosomes, or your 23 pairs, all right, 23 of those came from the mother and 23 of those came from the father. All right. So you could see that if we had a cell with 23 pairs of chromosomes fused with another cell with 23 pairs of chromosomes, we would have way too many chromosomes. Ugh. Okay, so a couple terms that we kind of need to understand before anything else. A haploid cell is a cell that has half the amount of DNA, as it should. All right. A diploid cell has two pairs of each chromosome. Or a diploid cell has two of each chromosome. A haploid cell has one of each chromosome. All right, so it's often referred to as N. Diploid is often referred to as 2N. So there's two of each chromosome. So if you have 23 chromosomes, haploid has 23. If there's 23 chromosomes, a diploid has 2N, which is 46. 2N is what a human somatic cell has. Okay. Gametes are sperm and egg. All right, these are gametes. So gametes are cells, sex cells that have half the DNA. All right, they're usually N, they're haploid. A karyotype is a picture or illustration of homologous chromosomes. Again, homologous chromosomes is a pair of chromosomes as the same general type of information but may not have the same exact information. All right. Crossing over is the process that we'll see later where homologous chromosomes actually exchange pieces with one another, which increases genetic diversity. Tetrads are when, as we know during mitosis, when what happens in mitosis is chromosome number 1A will duplicate and form a sister chromatid, and chromosome 1B will duplicate and form a sister chromatid. These will line up separately as sister chromatids on the metaphase plate. But in meiosis, actually, this duplicate, 1B duplicate, and the 1A duplicate will all be attached together, and that will be called a tetrad. All right, a tetrad because there's 4. A zygote is what happens when a sperm and egg fuse. Now, thousands and millions of sperm will try and fuse with an egg, but only one sperm cell will actually fuse with the sperm cell. One sperm cell will only fuse with one egg cell, and the moment the first sperm cell fuses, the entire shell of the egg becomes impenetrable to any more sperm, which that's crazy how fast that happens because there's a lot of sperm going at that egg. The sex cells are cells that are haploid gamete cells that are created by the process of meiosis. Sex cells are created by meiosis. Sperm is the male sex cell and egg is the female sex cell. All right. Some differences than mitosis. It occurs in sex cells, not somatic cells. So only one group of cells in the entire human body have performed the process of meiosis. It is 2 cell divisions instead of 1. We're going to end up with haploid cells, cells with the half DNA they need. And we talked about that before because when a sperm cell and an egg cell fuse, 23 chromosomes are fusing with another 23 chromosomes giving the resulting cell the pair of the 46 chromosomes, which every cell in our human body has. And it forms gametes, not somatic cells. All right. So the process is pretty straightforward, with a few new terms. The few new terms are chiasmata. The new terms are tetrads. New terms are gametes, zygote, haploid, and diploid. So the same thing generally happens as mitosis in the beginning. We go through all of interphase, we duplicate our DNA. One of the differences, though, is by the time our DNA condenses by prophase, all right, our sister chromatid pairs have attached to one another, all right, to form tetrads. So what you could imagine, if we were to go back here, is that if chromosome 1A here duplicated and chromosome 1B duplicated, those four chromosomes together would be called a tetrad. So you'd have a duplicate 1B and a duplicate 1A. Now what's going to happen with these is these duplicates are going to cross over and exchange pieces. All right, sometimes once, sometimes twice. So this chiasmata, all right, is the place where the crossing over occurs. Now this crossing over occurs by an enzymatic process of cleavage, which means that an enzyme will cut the piece of DNA and exchange it with the exact piece on the other chromosome. All right, on its homologous chromosome, remember. So it's not this red piece exchanging with this red piece, it's this red piece of this sister chromatid, this red one, exchanging pieces with the other chromatid. Now remember, this blue one has the same type of information as the red one, but it doesn't have the same exact information. It has variations or forms called alleles. So the crossing over, what it's going to do is it's going to result in these pieces having pieces of the other homologous chromosome. Now what happens is, at metaphase, again, so everything else is the same. The nuclear envelope is breaking down, the spindles are moving to the poles, centrioles are there. We go to the metaphase plate. Now the tetrads are lined up at the metaphase plate, and again they get pulled to the poles during anaphase. All right. Now again, what's going to happen during telophase? All right. Telophase and cytokinesis is we have the cleavage furrow, we have the partial formation of the nuclear envelope again, but the nuclear envelope isn't going to entirely reform, and the sister chromatids now, they're not tetrads anymore because they're not attached, are not going to unravel to form chromatin like they would in mitosis. What happens is these cells are now going to go directly into another cell division called meiosis II. Now meiosis II, everything's the same. Our nuclear envelope will partially reform, but it's actually breaking down again at this point, just as in prophase 1. Our spindle is going to the poles. Metaphase, we line up at the metaphase plate. In anaphase, we're pulling them apart. So the moment we're pulled apart here, we have anaphase. And in telophase 2, our DNA is going to start to, we're going to have our cleavage furrow. The cell plate will form in a plant cell. Because remember, plants do produce egg and sperm as well. And our nuclear envelope will begin to reform. This DNA will go into chromatin. Our spindles will begin to degrade. And we will end up with cells that have half the amount of DNA as a normal somatic cell. So what we start with is a somatic cell, all right? And what we end with is 4 cells, all right, that each have half the DNA that's necessary. So we start off here with one set of 23 pairs, and then we duplicate to become 46 pairs. Now these 46 pairs divide, and now each of these 2 cells has 23 pairs. Now when we go through another division again, instead of having 23 pairs, each of these new cells will have 23 total chromosomes. So that when fusion occurs, this cell with 23 of each chromosome 1 through 23, all right, will fuse with another cell that has 1 through 23 of a chromosome with number of chromosomes again, but they will now fuse, and their chromosome number 1, all right, from the sperm cell will fuse, not fuse, but kind of stay near the chromosome from the egg cell. Yeah. Oh, wow, tired. Sorry about that. So that's the process. So some differences between the two. Mitosis is one cell dividing into two cells. The resulting cells are diploid, and the starting cells are diploid. In mitosis, we have one cell division. In mitosis, we're producing somatic or body cells. In mitosis, crossing over does not occur. In meiosis, one cell results in 4 cells. The resulting cells are haploid, not diploid, because they're designed to fuse with other sex cells. There's 2 cell divisions, not 1. We produce sperm and egg cells here, not somatic cells, and crossing over occurs, and the crossing over does not occur in mitosis. So this is kind of just a crash course on meiosis. We'll talk about it more in class on tomorrow. And we don't have class on Monday, but we do have class on Tuesday. We will be, I will be presenting some sort of evaluation for mitosis and mitosis for you that will kind of go through Friday of next week. So, yeah, if you have any questions about this, please look it up more on YouTube. You can ask me questions more as well. And I'd like you to kind of record some questions that you have on what I've just said, because this process can be confusing. I actually didn't quite understand the process of meiosis in high school. It wasn't until college that I actually really understood the process. So, what's going on?